So good afternoon and welcome today to a new time, only for today at 1 o'clock. And I am Audrey Russo. I'm President and CEO of the Pittsburgh Technology Council, and welcome to Business as Usual. I'm very thrilled by today's guest, and I will introduce him in a moment. A couple of things before we start the call. Thank you to Huntington Bank for the work that they've been doing in supporting us, as well as driving the work around the stimulus and getting people what they need from this SBA. And they have provided tremendous support over the last few weeks. I also want to thank the team at AT&T for their support in our public policy series, and more importantly, for the support that they provide the entire community during this pandemic. They had announced 3 months of free wireless service for frontline nurses and physicians nationwide at FirstNet Network, and built a partnership with a public-private partnership with First Responder Network Authority. So they have also additionally provided support for organizations focused on food security, and most recently 412 Food Rescue. I do want to say we have a packed week this week. Tomorrow we have Chris Johnson, President of Teletracking. He's going to talk about the work they are leading and accelerating during this unusual time. We also have Leslie Davis, Executive Vice President and COO of UPMC. And then later in the week, we have Senator Casey, who will be joining us from his home office. So packed week as usual, but I want to jump right into this show. But before we do, I want to remind you about the technology. We have put all of you on mute just so that we can eliminate any background noise that most of us have. And we will ensure that you have a chance to actually ask questions. So we have a chat box that's at the bottom of the screen. And we would like you to just put your name and the title of your company, and we will hopefully get to some of those questions. And we like to make this interactive. And if there are questions that we haven't gotten to, we will work with our guests today to see if there's any information that we could actually put out as we archive this. We record everything and we archive this. So today's number 21, and I'm thrilled to introduce Dr. Louis Falo Jr. He's an MD and PhD, and he has lots of titles and lots of experience. So on that note, Dr. Falo, why don't you just tell us a little bit about your background, and we're going to jump into what you've been up to.
Sure. So thanks for having me on. I'm a local guy, so I was born here and grew up in Greensburg. I went to Pitt as an undergraduate student. From there, I went to Harvard Medical School, where I did my MD training and also PhD training. Was very fortunate there to work in a laboratory of Dr. Benassaraf, who was a Nobel Prize winner and specialized in the area of antigen presentation, which is directly relevant to vaccine development. I did my clinical training all in the Harvard system at Mass General. And then after being on faculty at Harvard for a couple of years, I came back to the University of Pittsburgh. I've been chair of the Department of Dermatology for a little over 20 years, and also running a very active laboratory program that's been focused on skin biology, skin immunology, and in recent years, the last 5 or 6, bringing bioengineering into the area of skin immunology and drug delivery. And so that's the perspective that we had when we started down the road to this project.
Very exciting, very impressed, and very glad that you're back in Pittsburgh. So it's nice to see your career come full circle here and be one of the ambassadors for the future. So let's start with the University of Pittsburgh was one of the first groups to release promising peer-reviewed results in the pursuit of vaccines to combat SARS-CoV-2, the new coronavirus causing COVID-19 pandemic. Can you start out by talking about those early results, but also talk about the journey that your team has been on in the recent years that allowed Pitt to react so quickly?
Yeah, so this is really a collision of 2 evolving technologies, both of which we've been developing here at Pitt. And so I have a very close collaborator in this work, Dr. Andrea Gamboto, And he has been focused on this antigen that we are using for this vaccine, which is called the S1 antigen. You may have heard about it in the news. It's a well-established target for coronavirus vaccines. So Dr. Gamboto was working on this S1 protein for coronaviruses, different coronaviruses, back since 2003 when he was studying SARS and then later on MERS, and working on efforts to develop a vaccine at that point in time. He was able to craft several genetic constructs that made it easy to express this protein once we got the sequence that was released by the Chinese in mid-January. What happened was we were very able to put that sequence into those constructs and do work with this specific virus. The paper that you were referring to describes this virus and this vaccine in the context of previous work on the other viruses. And so we show data on how this particular vaccine works with the earlier coronaviruses and then compare it to how it works with the new coronavirus. And then the other technology that collided with this is the development of the delivery platform. And so the delivery platform is really a platform technology. It's designed to deliver drugs, vaccines, other agents, very specifically to microenvironments in the skin. And so we've used this platform for multiple purposes, including skin cancer treatment. And in this purpose, we're adapting what we've done previously with vaccine work to put the protein construct that's specific to the coronavirus into this platform. And
so you've been working on this since when?
So the platform's been under development probably for 8 to 10 years. So we have a long history of working on ways to get these drugs into skin, and in particular, the microneedle approach.
And so for the last 2 months, you have a team of how many people that are actually working on this?
So altogether, we have about 11 people who are working. And I think that and that includes some people from Gamboto's laboratory. And you know, the nice thing is that it spans a really broad range of people with expertise in immunology, skin biology, and bioengineers, all working side by side.
And everyone's keeping safe, right? I would imagine that they are.
Shift work. So we have everybody else is gone. So we've actually got lots of room, and people work in shifts in the event that someone would actually come down with a positive result, and that whole team would then need to come out and be quarantined. We'd still have another team ready to go.
Okay, that's great. So Pitt's approach doesn't just involve a new vaccine, it includes this delivery system that you mentioned. Can you talk about that more so people can understand what that means?
Sure. So the delivery system it works essentially just like a Band-Aid, except it's a Band-Aid that instead of having gauze in the middle, has a 400 small microneedles. So the microneedles are each about the width of a human hair and about 0.5 millimeter in length. So what happens is these microneedles are basically made out of a sugar substance. We take the protein antigen, and this is a traditional protein approach, just like a flu vaccine, and we mix that antigen in with the sugar and harden it. And so when these needles and this Band-Aid is applied to the skin, the needles are in a hard state and they puncture through the outer skin layers. But then once they're in the skin, they very rapidly absorb fluid and completely dissolve. And so the needles are the vaccine. And when the needles dissolve, they release the vaccine into a very specific area of the skin that's very good at generating immune responses. And so that's basically the technology in a nutshell.
Okay, let me see if we have a question here that I might want to jump to. What's your thoughts on the proposal? This is from Ken Chiachia, and he's saying, what are your thoughts on the proposal that a vaccine trial include challenging vaccinated individuals with the real virus?
So I don't know that anything like that would be necessary under the current conditions. So there are some areas, both of the United States and across the globe, where the infection rate is relatively high. So you could imagine doing a large study where you vaccinate groups of patients, and there would be some natural infection rate that would normally be occurring in the population, and you would compare the vaccinated individuals to individuals who were unvaccinated just in the normal population. So I don't think a challenge type experiment would absolutely be necessary in this situation.
And so who else is working? Are you working with anyone else outside of the University of Pitt on this in terms of
Yeah.
your multidisciplinary team?
Yeah, so we have a collaborator in the Netherlands who happens to have models that are able to look at the antibody responses regenerate and evaluate whether or not they actually bind to the virus.
And so what do you think the time frame is for this?
Yeah.
If you had sort of your magic wand.
Yeah, so that's a really difficult question to answer, and I'm never being evasive about this, but it's because we don't actually know. So the process of developing a vaccine is an iterative process with the regulatory agencies, and so what happens is you gather up all the information and data that you have, you submit that in the form of an IND, and then you begin a process with the FDA of back and forth. You know, they may want another experiment, they may want more information. We do that, we give that to them, and then we see where we go from there. And so it really depends, when we actually start the trial, it really depends on that interaction and what we're gonna need in addition to what we have. And it's not, you know, it's not a horrible thing that this process takes a long time, because the most important thing is that we have a safe vaccine.
Right.
And the FDA, that's their job, and they do a great job at it. And so, you know, I think that, you know, we need to be patient with this, not just for this vaccine, but for all the vaccines in development. Once trials start, again, there's wiggle room in the timeline, and everything is very carefully monitored. So typically, as Dr. Fauci has been saying, a year to a year and a half. This can be sped up, and there's a lot of people at the FDA working hard to do that. But that also depends on the results of the trial. So if the vaccine in the beginning is safe and effective, we can go quicker.
So here we have a question. I've read that there's a dermatological issue with those who actually have the virus. Is that true? And this is from Douglas Holmes. He's CEO of Dermatologist on Call Diagnosis.
Yeah, so there have been some what we call case reports of cutaneous manifestations, rashes, et cetera, associated with people who have the virus. Cause and effect there is very difficult to determine from case reports. And so, you know, it's basically anecdotal at the present time.
So do you think, following up, comparing vaccinated folks to unvaccinated folks, how important is the effect that vaccinated people in your trial may behave differently because they have the belief they're probably protected? Is there a placebo group important in vaccine trials like this?
There is, and the placebo can either be a group of people that get something, for example, a microneedle array that's empty, or it can be a control group that's structured from people who have no treatment at all. And so just part of the population that you follow that match the characteristics of the population that you've actually immunized. Now, in the trial stage, it shouldn't alter behavior, because patients are well aware that it's a trial and that we don't know that it's an effective vaccine. I think, again, that's why it takes so long, is because we would like to be sure that when a vaccine is actually released to the public, that it's actually protective, because if not, there could be false confidence.
Right. And so where will you do the clinical trials, do you think, and when do you think they'll begin? And I know that's sort of a tough question, because you mentioned the FDA process.
Yeah, so everything's evolving. We would love to do the clinical trial in Pittsburgh. That's our goal right now. But again, it's evolving, and we'll have to see how everything works out.
Okay, that's great. So let's see, most people want to know what comes next. So what are the steps that need to happen before the vaccine can reach the population and help, hopefully, to bring about the demise of COVID-19?
Right. So we go through the process of getting approval to start a clinical trial. Clinical trials start with what we call a phase 1 trial, which is a small group of patients, maybe between 40 and 60. And the goal of that trial primarily is safety. So patients will get vaccinated, and they'll be followed very closely for several weeks to see if anything bad happens. Once we're to the point of showing safety, this same group of patients will be monitored for whether or not they're developing antibody responses. If things are very positive and look good, we will expand that into what they call a phase 2 trial, which then goes to hundreds of patients, and you start to look at real efficacy as measured by the amount of antibody that gets produced and how effective that antibody is at binding to the virus. After that, you go into really large groups of patients in a phase 3 clinical trial, and that's the last step that's necessary until you actually are licensed by the FDA to produce and sell the vaccine.
So here's a question from Tom Fry. He's asking, has the virus been reported to have mutated to any extent that it's potentially delaying any of the trials?
Yeah, so this is an interesting question. So unlike flu, which you all know mutates constantly, and we're always chasing the antigens on the flu virus every year, and that's why we need a new vaccine every year. What we know about previous coronavirus infections, MERS and SARS, is that the coronavirus tended to be very stable in those diseases. So far, it's too early to tell, but we don't have any indication that there's any significant change in mutation. And part of that is because the protein that we target in this is an absolutely essential protein for the function of the virus. It's the protein that the virus uses to bind to the receptor on human cells. And so if that were to mutate, we would guess that it would be more difficult to bind and the virus might not be infective at that point.
That's interesting. So let's talk a little bit. First of all, we're very proud. I mean, just for you to be here in Pittsburgh, and we've got a ton of questions here. That, for example, Sean Hamill is asking: are you planning on testing the vaccine in monkeys first and challenging them? And if not, why?
Yeah, so one of the difficult things about this vaccine process, which is moving ahead very rapidly with great urgency, is that those models are just being developed now. So even the animal models haven't really been developed and validated. And so we're basically looking at the effectiveness of vaccines in generating antibody responses. And then that's where previous work comes in, where our group and others have been able to show that those types of antibody responses are effective in these larger animal models against the virus. So it's kind of a connection to the old with a slightly different virus as we try to race ahead with the development of the models to be able to test this in non-human primates.
So is it a possibility that the vaccine will not be needed with the way the number of tested and infected people continue to increase without the death rate following? There's a whole bunch of models there.
Anything's possible, and all the models are models. We see the models are changing every day. And in fact, for MERS and SARS, the vaccine development was greatly hindered by the fact that the disease kind of went away. And so the impetus to spend more money on getting these MERS and SARS vaccines out there went away with the disease. I hope that we learn from that, right? If we had licensed vaccines for those two diseases right now, it would be relatively straightforward to test them for this virus, especially if they targeted similarities in the protein structure. I think we need to be aware that these efforts, and these technology platforms that we're developing for vaccines need to be developed on an ongoing basis. So even after this is over, there's a good likelihood that there will be another pandemic. And if we don't have these tools already in place, it takes a lot longer to respond.
So here's another question from Griffin Schultz. If I understand this correctly, in addition to potential vaccine, you have a novel way to deliver the vaccine. That's the Band-Aid apparatus. So is the Band-Aid apparatus just another way to deliver the vaccine that's less invasive than a regular needle, or is there the additional benefits as you articulated earlier?
Right. There are actually several benefits. So the first benefit is that it's very effective because it targets the skin immune system. And so if you think about it, your skin is your outer layer of defense, and every day you're getting bombarded with viruses, bacteria, et cetera. You don't even know it because your skin does a good job of protecting you. Because of that, the skin has evolved to be very immunogenic, more immunogenic than muscle, which is where most of our needle injections end up for vaccines. And so that's one advantage. The other advantages are, and this is great for patients, no pain and no bleeding. So the Band-Aid feels a little bit like Velcro. You only need to wear it for 5 minutes or 10 minutes at most, and then you tear it off and throw it away. There's no real medical waste involved. So it's a very safe delivery process. And then perhaps the game-changing advantage is that this technology eliminates the cold chain. So when we're talking about vaccines, most all vaccines need to be either frozen or refrigerated from when they are made until when they go into the patients. And so this creates a very expensive challenge for storage, delivery, and logistics. The microneedle arrays, once the antigen is incorporated into the sugar matrix of the microneedle array, it's stable at room temperature. And so these vaccines can be put into boxes, stored, shipped, just like Band-Aids. So for global immunization programs, in a lot of places in particular where there isn't good refrigeration systems, this is really enabling in terms of beginning effective global vaccine campaigns.
So here's one question. I don't know if this, it says, any thoughts on the comments by Dr. Gallo? Do you know Dr. Gallo on this oral polio vaccine as a solution?
Yeah, well, an oral vaccine is great as well. So oral vaccines have that same advantage of being relatively stable and really easy to deliver. You just hand out pills. And so, people working on that, that's one of the types of vaccines that eventually will get into clinical trials. The best thing about this is, we're seeing a new level of collaboration among scientists all over the world. So collaborating within institutions, but across institutions and across the globe. And what we're going to see, even with multiple interactions with the pharmaceutical sector, we're going to see 10, 20, 30, 40 vaccine candidates progressing into clinical trials. And that's a terrific thing, because the more that we have getting into clinical trials with different technologies, the better the chance that we're going to get the right vaccine, and the possibility that we have technologies that emerge that are better in general than what we have now for vaccines, and will change the way we do all of our vaccines.
Mm-hmm. So here, this comment is about that sugar is the magic of mRNA technologies. So that sugar is nature's great molecular stabilizer. Any other vaccine candidates using sugars to stabilize antigens?
I am not directly aware of that. It would not surprise me if there were, but I know for the microneedle, and particularly the dissolvable microneedles, that's a great matrix.
So here's a question from a 10-year-old on the call.
All right.
Hi, I'm 10. How long should this take?
This should this should be ready soon enough that once this episode of the pandemic goes away and we're all able to return to work, we will be able to eventually have this vaccine so that this doesn't happen again. So this is not for what's happening right now. This is for what's going to happen or could happen in the future. So we don't ever want this to come back.
Thank you for asking that question. Excellent. So what can people in Pittsburgh do to support your work? Do you see any role for the tech sector to play that would be helpful to you and your team?
Yeah, so with this current vaccine, as you might imagine, we've had a lot of interest, and I think we're pretty well resourced at the current time. The really important thing is to be able to sustain a program for vaccine development, something that would enable us to have predetermined FDA approval for delivery technologies, et cetera. And so our goal is to establish a center or some kind of organized structure for continuing vaccine development and bringing together skin biologists, immunologists, and engineers to work on these technologies and perfect them and get them approved by the FDA.
So when do you think we should check back in with you?
So I would say when you hear the announcement that we have begun clinical trials. So, hopefully that won't be too far off, but that's the next really big step, and something that I think will be important to all of us.
So we have spent almost 30 minutes with you. You have been incredible, and I've tried to ask almost all the questions that are here. We take a lot of pride. You are talking to a lot of smart people, as you can tell by the questions that were asked. And we want to do whatever we can to help you, help tell the story. And is there any, here's one last thing, is there any procurement coordination steps being taken to prepare for mass manufacturing and distribution of a vaccine when it's created to keep up with the demand, or are we getting ahead of ourselves?
So we're getting a bit ahead of ourselves. We do have a business development team at UPMC through UPMC Enterprises that is helping in those efforts. And I think the commercialization pathway is being developed as we speak.
Okay, is there anything else you want to say to us, all of us?
Just thanks to everybody for hanging in there with the social distancing, which is what we can all do right now to help the situation. And I think that we should all have great hope. There's a lot of progress, not just in this, but in therapies, other vaccines. We're going to win against this virus. And, you know, I have great confidence that we're going to be able to get everybody back to work in the not so distant future. So
you think we're doing a good job in Pittsburgh?
I think we're doing a great job in Pittsburgh. I mean, we have done a great job in keeping the incidence low compared to other cities, especially our neighbors to the east. And, you know, as I go out and about in town, I see a lot less people than I used to see, and I see people doing the right thing all the time.
That's great. I can't thank you enough. We're going to keep tabs on you if that's okay.
Sure.
Stay connected and do what we can to make sure that Pittsburgh and the world knows what you're working on. My hat's off to you. Please be safe. We need you, and we need your team. And thank you for your leadership across an interdisciplinary team. So everyone, we are signing off. We actually, this week, I mentioned earlier, we have some great guests coming up. We have tomorrow, Chris Johnson, who runs Teletracking. He's the president there. We have Leslie Davis on Thursday, EVP and president and COO of UPMC. And Friday, we have Senator Casey. So thanks, everyone. And until tomorrow at noon.